So a very short introduction to our company for those of you who are new here. Metabolon is the global leader in metabolomic services. For over 20 years now, we have developed unrivaled capabilities in metabolic profiling, really setting the standard in terms of data quality and metabolite coverage, metabolite annotation, particularly in untargeted metabolomics, which is of course facilitated by our reference library of standards, which we have built over the years. Metabolon is committed to advancing the field of metabolomics and transforming our understanding of human biology and human health and disease. Just a little bit of context to today's webinar. So we know that complex diseases such as cancer, cardiovascular diseases, metabolic diseases, they all arise from a multitude of genetic, environmental, as well as lifestyle factors. We know that traditional single omics approaches, which focus on a single level of molecular information, they often capture only a very small part of this intricate interplay between different biological layers. A multi-omics approach, so integrating data from genomics, transcriptomics, proteomics, and metabolomics, provides a more comprehensive picture of the molecular landscape of complex diseases and gives us a more multidimensional view of human biology. And this is, of course, facilitated by the implementation of advanced bioinformatics and computational tools, in parallel, of course, to experimental validation and subsequent clinical translation. But why do we focus on metabolomics? Why do we think that the multi-omics puzzle is simply not complete without a deep dive into metabolism? Metabolomics really provides a snapshot of the entire physiology of a biological system. Metabolites, as they are positioned in the central dogma, are the downstream products of the cumulative effects of the genome, the transcriptome, the proteome, but also the environment. We often like to say that metabolomics sits the closest to the phenotype of all the other omics fields, as it directly measures the end products of all the cellular processes that happen at every at all different levels of biological organization. So metabolomics is best positioned to describe the ultimate molecular phenotype, which Ania is going to be talking about more in her presentation, making it an ideal tool for biomarker discovery and validation, as well as other aspects such as the identification of novel drug targets, advancing our understanding of disease mechanisms, and so on. It can, of course, be used as a standalone approach or in combination with other omics. And we can leverage metabolomics to gain additional biological insights, which might not be captured by technologies such as genomics or proteomics. Of course, metabolomics does not negate the need for other omics, but it can bring an immense added value to multi-omics research. While the genome illustrates, of course, an individual's risk for a specific disease based on their genetics, their metabolome provides a functional readout of the actual current state, healthy or disease, providing therefore a definitive representation of the phenotype. And lastly, just wanted to touch upon some of the challenges that come with multi-omics. Multi-omics research offers great potential, but we're still far from getting there. There's a lot of obstacles for us to overcome. Integrating data from different omics platforms, which come with heterogeneous formats, different scales, different measurement techniques, and so on, present a very significant analytical and computational challenge. We, of course, need to normalize and standardize the data across different omics data sets in order to achieve meaningful integration of data and comparison across data sets. The sheer volume of data that comes from these multi-omics exercises requires very advanced bioinformatics and statistical methods to manage, to analyze, to visualize the data, and ultimately successfully achieve biological interpretation. And here I really want to highlight how crucial the interaction between bioinformaticians and biologists is in this case. Understanding this complex interplay between the different omics layers and the implications that they have in disease development, disease progression, treatment identification really cannot be realized without this synergy. And I think this is going to be very apparent in Ania's talk, together with, of course, data sharing and collaboration, which is essential for accelerating multi-omics research. And again, Ania, I think it will provide some more information as to the steps they're taking towards making multi-omics data more accessible. And finally, of course, bridging the gap between multi-omics and between research and clinical applications. For that, we of course need more translationally relevant tools, and of course, validating multi-omics findings in the clinical setting, translating them into effective diagnostic and therapeutic strategies will be key towards unlocking the potential of multi-omics research. And with that, please join me in welcoming our speaker, Dr. Ania Halama, she's currently working as an assistant professor of research in physiology and biophysics at Weill Cornell Medicine-Qatar. She earned her PhD from the Technical University of Munich in Germany in 2013. And since then, she has been working in the field of metabolomics for about a decade. Her research focuses on the metabolic dysregulation in complex diseases, particularly cancer and diabetes. She has a very successful track record in the integration of metabolomics with other omics technologies for clinical applications, particularly through population health studies, which will be the focus of her presentation today. We're very excited to hear from her and see all the great work that they have been doing together with Karsten Sucher and their wonderful network of collaborators. Ania, the floor is yours.
Thank you, Natasha, for this kind introduction, for having me here. I'm right now sharing the screen. Perfect. So good morning and good afternoon, everyone. It all depends from where you are coming. So in Doha right now, we are having 5 p.m. and the weather is still very good. So sharing a bit of sun with you. Thank you, Natasha, for making a stage, in fact, for the talk, which I will gonna be focusing right now. So before we are going to the details, I just like to say that there is no conflict of interest which I need to disclose. So as Natasha was saying very rightfully, that there is increase in the interest in omics research, and now it could be asked why it's so. So basically, the physiological as well as pathophysiological processes in the human body can be nicely described with clinical chemistry, and this is something which we used to, and molecular markers related to different diseases which can be captured with clinical chemistry are pretty known to everyone. But unfortunately, they are limited in their capacity to really capture complex diseases and identify the processes which might be more optimal for patient stratification and treatment. And thus development of genomics, also epigenomics, transcriptomics, proteomics, glycomics, and metabolomics, which are all omics layers, provide further insight and is giving us more information and better characteristic of human pathologies and also physiological situation. So before I will gonna be diving into the multi-omics layers, and I will introduce you to the molecular human, which is part of this presentation, I would like to focus shortly on the metabolomics and metabolome, and also how we use metabolomics as a tools to answer several of our questions. So for those who are not very familiar with metabolomics, the term metabolome is with us since around 25 years and is really capturing a small molecule composition in given sample. And if you are thinking about sample and what kind of samples can be measured using metabolomics, as you can say that the sky is the limit only, because we are able to implement metabolomics in different biofluids, including saliva, urine, plasma, but also tears. A recent study were showing that air breath, it could serve as great matrix for metabolic profiling. Also, it can be implemented to tissue samples and cells. So I will gonna be focusing today mainly on biofluids from human, and it will be saliva, urine, as well as plasma. So I would like to start with the Qatar Biobank study and metabolomics, which we conducted here. So just brief about Qatar Biobank. So Qatar Biobank, like any other biobank, is focusing on the enrollment of the population, in this particular case, population of Qatar, as well as expats, which are residing here since more than 15 years. So right now, the enrollment is still, of course, in process. There is around 38 individuals which were enrolled to Qatar Biobank. If you are more interested about Qatar Biobank and the processes which they have in place, please visit the website, which is visible just here. So from the Qatar Biobank, we were obtaining a subset of 6,000 subjects, out of which 3,000 we could submit for metabolic profiling. What is important here, the samples which are measured, which are taken from Qatar Biobank and Qatari populations cannot go out from the country for the measurements. So in order to conduct metabolomics measurements, we brought the platform from Metabolon and we installed it in anti-doping lab here in Qatar, and Weill Cornell Medical College, together with two other institutions, QBRI and also Hamad Medical, were supporting ADLQ in bringing Metabolon platform to Qatar. This platform was used for the measurements of the 3,000 subjects from QBB. Also, for the same individuals, for 6,000 of them, we were having questionnaire, and those questionnaires were reflecting on the drug usage by that particular people. So why this is important from different perspective, especially if you think from the epidemiological point of view, you like to know what kind of drugs your population is really relying on for future. It's pretty important. Yet it's important to say that not necessarily all responses to questionnaire asked are accurate, and there might be several biases. This is the reason why we thought whether or not we can use metabolomics to validate the questionnaire answers. So before we go to the metabolomics measurements, I would like to just shortly introduce you to our population of those 6,000 QBB participants. The BMI and HbA1c levels were measured, and everything else were from the questionnaire or the documents provided by the participants. So what you can spot here, our population is pretty young because the mean of age is 39. Also, if we are looking from the perspective of BMI, we are having slightly overweight population where the BMI mean is 28, almost 29, and a little bit enhanced values of HbA1c. If we look at the population generally, around 17% of people is having type 2 diabetes. So what you can see here, it's the answers to the questionnaire, and in those questionnaires, people were asked about whether or not they are treated for diabetes with tablets, with insulin, but also the questions were reflecting on multiple other diseases. So among all, when we were looking at least one over-the-counter or prescription drugs which participants were taking, so around 51% were saying that indeed they are on some medications. So what you can see here right now is the representation by sex, females and males, and their age. Again, our population is young, so the majority is individuals between 25 and 34 years. What we asked as a first, we wanted to understand whether taking regular medications is kind of correlating with the age or progression with in age, because it would be kind of positive control for the data and information retrieved from the questionnaire. And what you can see here on the plot, there is indeed correlation between the regular medication which has been taken with the age of participants, and this is true for both females and the males as highlighted here. We also look further to understand what is the majority of drugs which the participants are taking, and we found out that A10 group, which is from the ATC therapeutic subgrouping, and also C10 are the most prevalent drugs which are taken in the population. So A10 and C10 are drugs used in diabetes and also lipid modifying agent. So we looked at the frequency of those drugs are taken, and so what you can see here, they are increasing over time, and more or less the distribution between females and males is similar. Also, when we looked on the thyroid therapy, which thyroid diseases are more frequent in females, and there is in fact 10 times more females suffering of thyroid diseases, you can see first of all that indeed it's kind of confirmation, more females than males are on this medication, and also there is certain increase with the age. So this was kind of positive control of the data which we're obtaining from QBB. So now what you can see here is the list of the drugs which we were looking at. I mean, of course, the list is longer, but this is only an example. And the number of subjects which were self-reporting on taking of those drugs, and versus the number of those drugs or the products of their metabolism which were identified across of those individuals. So in fact, the actual comparison you can see here, where there is number of drugs which were only self-reported or only detected in the blood, or detected in both, so self-reported and detected in blood. So for majority of them, or many of them, self-reported kind of was matching with the one which we were seeing in the blood, but we are having also multiple examples, like for instance, the one with acetaminophen, where the number of self-reported was absolutely not matching with the number of the one which are detected in blood. So we were identifying acetaminophen products of metabolism in over 400 subjects, and it was not really matching with this, what was reported by them. Also, another kind of, in our view, interesting story was the metformin, which was detected as in over 600 individuals, but only self-reported in 220, and this was overlap between both in the number of 200. So of course we scratch our head and we try to understand what's going on here. The good story about acetaminophen, it's a drug which is metabolized in our body, and this is happening via three main pathways: CYP, SALT, and UGT. So each of this pathway is resulting in formation of different product of metabolism of acetaminophen, and all of those were detected in blood. So what we are trying to understand, whether or not the data which we are obtaining are kind of false positive, or there is other explanation to it. And what you can see here, there is certain overlap between the individuals which were identified. So there is a overlap with the metabolites, product of the metabolites across the individuals, which is suggesting that in fact those individuals were taking the medication but not reporting on it, or maybe they were not even considering acetaminophen as a drug. So suggesting that acetaminophen usage is mainly or likely underreported in QBB population. So in fact, the richness of this data led to further study, and this was done by Gorev, who was looking specifically on the new pharmacogenetic loci which are related to acetaminophen. I won't gonna be talking about this one today, but if you are interested, I strongly encourage you to look at this paper, because he was able to identify pretty interesting story there. So now going back to metformin. So in this particular context, the story is more tricky, because metformin is not metabolized in our body and is just excreted. So it's not so easy as in case of acetaminophen, when we can really find out if this is a problem of one molecule which is wrongly annotated or carry over. In this particular case, unfortunately, we are not having such a luxury. But knowing the human nature and also the way who is really lately taking metformin, we know, for instance, that multiple people which are overweight or obese, they are relying on metformin to lose some weight. There are also others which like to normalize their HbA1c level, and they are also taking metformin. And even patients which are changing the diet because of the diabetes, but they are not reporting on the actual medication usage. So we were trying to understand whether inclusion of this and trying to overlap those with metformin, which is detected, will gonna help us to solve the issue. But unfortunately, we were not lucky this time, and we were still seeing around 360 metabolites which were detected in blood in the individuals which were unlikely to take metformin. So we conducted this story further, and we restricted our cohort only to participants which self-reported on taking metformin, and they are here highlighted in blue, and the one which were unlikely to take metformin, meaning young, low BMI, and low HbA1c level. So what you can see here in participants which self-reported on taking metformin, there is elevated level of the drug which can be found in comparison with the group which is unlikely taking metformin. Yet still, in the participants unlikely taking metformin, there is signal of metformin. So we engaged in this particular study strongly with Metabolon because our first thought was that in fact here is a carryover between different samples. But the colleagues, especially Ania Evans from Metabolon, she was conducting additional experiments to prove that there is no carryover possible. So, of course, the question is, what would be possible in such a scenario? There are multiple different strategies. One of these could be that there is another molecule which is truly similar to metformin, which we are capturing, or the individuals are kind of exposed to metformin. But this is something which we were not able to really identify further. So more about how metabolomics was used and more about the drugs which were identifying, which was not necessarily matching with the self-report of medication taken in QBB, you can see in this manuscript, and I'm strongly encouraging you to have a look if you are interested. So now I would like to change slightly gear to talk about how metabolomics can be used, or metabolism can be used as a potential signature of different diseases. So if we are thinking about the differences between healthy and type 2 diabetes, we know that there are very solid markers which were developed over years, which is glucose, HbA1c level, as well as 1,5-HG, which are fairly well discriminating between healthy and type 2 diabetes. Also, the massive amount of the work which has been done in context of type 2 diabetes and metabolomics resulted in identification of metabolic markers such as branched chain and aromatic amino acid, mannose, and elevated lipid levels, which are known to be characteristic of type 2 diabetes. But type 2 diabetes is preceded by the insulin resistance. So if we are thinking about insulin resistance and how insulin resistance is determined, the golden standard here is monitoring of hyperinsulinemic euglycemic clamp, or HOMA-IR. So this is monitoring for hyperinsulinemic euglycemic clamp is pretty invasive method, because you need to put your patient into hyperinsulinemia and then you are monitoring the amount of the glucose which needs to be injected to reach normal glycemia. Thus, further study has been conducted, which were looking at the metabolic profiles of individuals with insulin resistance, and also those were established. Important here in terms of the identification of insulin resistance or having markers for insulin resistance is that this process of progressing towards type 2 diabetes could be reversed. So it's good to have molecular markers or metabolic markers which can identify the process before. But what we were trying to think about, if we are talking about insulin resistance, which is already pretty far progression towards the disease, and type 2 diabetes, then our question was, is there something which is preceding even insulin resistance? So we were thinking about, is there any specific characteristic of the low insulin sensitivity individuals? So in order to answer this question, we were conducting study in collaboration with Professor Abu Samra from Hamad Medical Corporation, and he was recruiting 47 Arab males. All of them were young. They were having normal BMIs, normal HbA1c level, and normal OGTT. So technically speaking, they were normal, and you would never submit them as a patient for hyperinsulinemic euglycemic clamp. But to answer our question, which I was mentioning before, we proceed further with them, and we submitted them for this exercise with hyperinsulinemic euglycemic clamp. And for those individuals, the blood samples were collected after overnight fasting. Then the clamp was started, and after 2 hours of the clamp, the second sample was collected. So we were having 2 time points here. We divided the population of those males into 3 groups. The one which are identified as insulin with low insulin sensitivity, moderate insulin sensitivity, and high insulin sensitivity. And we try to understand whether or not there is specific association between those subgroups of insulin sensitivity and metabolic profiles. And to look at this, and this is the data which are coming directly from those individuals, you can see that they are indeed grouping in those 3 different sensitivity, insulin sensitivity levels, but those subjects are not different on the level of BMI and also HbA1c. So this shows that based on those 2 factors, you cannot really discriminate them. So for this particular study, we went for the metabolites measurement with global discovery panel of Metabolon, and also a complex lipid panel, lipidizer, as we are calling, to have like global overview on metabolic and lipidomic processes which could be found in those individuals. The first question which we asked was whether or not there will be metabolic differences before the clamp, so after overnight fasting, and at the moment of the clamp. And what you can see here on this PCA, indeed, there is a shift in metabolic profiles when we are looking at those 2 time points. We try to analyze the data to understand if we are really on the right track and if all the processes are correctly in the place. And what you can see here, we found that there is increase in the glycolytic processes which are occurring under the hyperinsulinemic euglycemic clamp, in contrast to beta oxidation processes which are present under the fasting stage, which can be observed after overnight fast. So basically, the differences between those 2 time points are showing us the shift between the utilization of energy, so from the beta oxidation under the fasting to glycolytic processes under the clamp conditions. So knowing that our data are in really good shape, we started our analysis, and we were looking for the associations between those 3 groups which we specified and the metabolic features. So what we were identifying, there was over 300 metabolites which were showing association with insulin sensitivity levels. And of course, we're very curious who is on the list or what kind of metabolites we see on the list. And what you can see here are majority of diacyl and triacyl glycerols. And in fact, majority of lipids were showing the differences. So there was association between the low insulin sensitivity and the specific lipids of diacyl and triacyl glycerols. We further ask question whether this pattern remained under the clamp condition, and indeed you can see that even under clamp condition, this fingerprint of elevated lipid metabolism in insulin kind of non-insulin sensitive individuals is true. So what we can see here and how we can conclude is that low insulin sensitivity is highly correlated with lipid levels which are elevated, so in terms of diacyl triacyl glycerols in the body, and this is independent on the condition or the body state of the individuals. Also, we propose that those D and triacylglycerols could serve as hallmarks of compromised insulin sensitivity, and in the future study, I believe those could be included in potentially in the measurements. Of course, we were further interested whether or not there will be some other metabolic features which can be observed under the clamp condition, and we found the whole list of different metabolic associations with different sensitivity levels of insulin. What was interesting here that in contrast to the baseline, under clamp condition, molecules such as branch chain amino acid and aromatic amino acid, as well as specific fatty acids, were showing to differ between the group. And we know that branch chain amino acid are already signatures of type 2 diabetes. So it's really showing that those individuals are compromised in terms of the responses to the insulin. So if you are interested in this particular piece of our work, please have a look on this manuscript. So this was a brief about how we can use metabolomics to answer these different questions. Of course, the list is longer, but for the interest of time, I would like to dive more into the omics layers and multi-omics, which we were discussing on the beginning. So if we are thinking about omics and multi-omics analysis, first of all, it's very important to have a cohort which will gonna have all of those measurements from the same individuals, from actually the same sample. And having that will gonna be letting us to talk about complementarity, also integration of omics, and we're gonna let us know how to analyze such a data and especially how to visualize them. This is the biggest struggle for me, seeing a lot of Excel sheets with p-values, which are not necessarily very informative. So here I would like to focus a little bit on those multi-omics and the approaches which we undertook to describe our molecular human. But before that, I would like to introduce you to the cohort which is Qatar Metabolomics study on diabetes. And this was the study which was conducted and initiated by Professor Surre together with Dr. Montgomery and Dr. Salim from Hamad Medical Corporation. It was the first study on diabetes and metabolomics in Doha, which were conducted. And in the framework of this study, we collected almost 200 controls and 200 type 2 diabetes patients, from which urine, plasma, and saliva was collected. Important here, and also important for whoever is interested in conducting multi-omics work, it's very good when you are aliquoting your samples right away, because you are avoiding the problems of freeze-down cycles. So this is only a side note. So we had multiple aliquots of those saliva, urine, and blood, and this is what we did with them. So we were having clinical chemistry data. We were conducting our genomics profiling using Illumina. Also, we look at methylation using Illumina. Gene expression profiling, we had RNA-seq with Illumina again, as well as microRNA using Hexagon. And proteomics was measured with two different technologies. One is Olink and second Soma, and this was done from blood. We also extensively look at the glycans. So we look at the total glycans and glycans, which are highlighted here as PGP and glycosylation of IgG. And those were done in collaboration with Genos and Gordon Lauk. Also, we look at the glycosylation of IgA. We further monitored lipoproteins using Nightingale platform, lipids using Biocrates P180, as well as Metabolon, the complex lipid panel. For urine, saliva, and blood, we were conducting the first generation of Metabolon platform. So it was Metabolomics, untargeted metabolomics profiling. For the blood, additionally, we were running on the second generation of the Metabolon platform, and urine was additionally monitored using Kenomics, and it was based on the NMR spectra. So overall, we were having like 18 platforms, which were covering 8 different omics, coming from exactly the same samples. So of course, we were using this data pretty extensively, especially to answer multiple questions reflecting on the diabetes signatures, but also looking for different GWAS associations and EWAS associations. And this is just a brief overview on the study, which has been conducted using our QM-DIAB study. Also, it's important to mention that frequently QM-DIAB is serving as a cohort for replication. So right now, having all of this data, which is over 6,000 molecular traits, and over 1 million of SNPs, 50,000 of transcripts, and almost 500,000 of CpG sites, we are in fact in the position to talk about the complementarity of the platform, the way of their integration, also analysis and visualization. Here is the overview on the experimental setting which we conducted. So first we look on the Gaussian graphical models, and this was done with the collaboration with Jan Krumzik, which is residuing in New York right now. So those Gaussian graphical models were used to understand the association within given platform. Further, we focus on mutual best hits, which I will gonna touch briefly upon in the next slide. Here we were identifying over 2,000 of mutual best hits, and this was for the first time this concept was used for multi-omics profiling. Next, we try to understand what is the association between genome and metallome, metallome and transcriptome, as well as transcriptome and genome. Additionally, we run multi-omics GWAS, multi-omics EWAS, and multi-omics TWAS, and so far we are aware of our TWAS were first truly multi-omics TWAS study. Finally, we are trying to understand further the phenotype and the way how different phenotypes is described throughout all of those platforms. So now maybe shortly about mutual best hits. So in fact, we were inspired by bidirectional best hits, which are frequently used in genetics to understand the orthology between genes coming from different organisms. And we use exactly the same approach to understand whether there is association or similarity between different platforms. So we were looking within omics between the platforms, and this serves us to answer more technical questions related to complementarity, but also the quality of the measurements. And we were asking more biologically relevant questions when we were looking more from the perspective of the associations across different omics. So what you can see here is the summary of mutual best hits which we were calculated, and they are highlighted here in the darker red part of the square. And how to read it? For instance, the number of mutual best hits between the two generations of metabolome platforms, which was generation 2 and 4, was showing us the higher mutual best hits levels, which was expected given that both are looking at the metabolic features. But what was important for us, we wanted to understand whether or not those mutual best hits are linking to identical molecules. And indeed, we were able to show that majority of those mutual best hits is linking identical molecules, highlighting further the quality of the platforms. We did exactly the same look through the Olink and Soma to see whether or not they are capturing identical molecules, and also for lipidomics platforms. Of course, we were looking at all of them, but I'm only highlighting those. Careful evaluation of those mutual best hits show us that between 72 to 93% of those mutual best hits is showing concordance between identical molecules which are detected at two different platforms, which further suggests very good quality of our data, and also the method which were applied in laboratories across the world. So now also very briefly about the biological relevance of the mutual best hits, which you can see here. So what I'm showing here is the mutual best hits which were identified between serpin A7 and thyroxine. So serpin A7 is thyroxine binding globulin and is serving as a carrier for different hormones to be delivered to thyroid gland. And as you can see here, the mutual best hits is really reflecting on true biological processes. We are having many of them, and when we were going to go further down the line through this presentation, you will be also able later on to use our resources to look at your molecule of interest. So we also implemented mutual best hit to look for further complex lipid structure and to reveal this one. So example here is showing the phosphatidylcholines, which are consisting of two different fatty acid chains, and some of the measurements are providing the total fatty acid chains in the as a measure, which is not necessarily very specific, and the actual composition should be revealed. And this was we were able to reveal this composition with the mutual best hits, where we were showing that those molecules are truly linked together. In fact, it was replication of the study which Gabi Kastenmuller and her team was doing previously. If you are interested, please have a look on her paper. But we took it a step further. We were also successful in showing or revealing the structure of more complex lipids such as thiacyl glycerol using our mutual best hit. And this is example again of the platform complementarity. So now moving to our GWAS associations. So we found around 700 of GWAS associations. Majority of them were reflecting of previously known associations, so we're replicating previous study. But for the one which we were adding the extra platform, in this particular case, it was EGA glycans. We were identifying new hits, which is ST3-GAL1, which is sialyl transferase, and this is contributing to enhanced sialation. It was shown that the level of ST3-GAL1 are resulting in enhanced sialation, so the protein. But what we are showing here, that in fact there is genetic variant which is contributing probably to this different profiles in terms of EGA glycans. And right now what we are looking at, we are trying to put them in the context of specific disease phenotype, and this is something which we are working right now on. Now looking on our TWAS associations, we found over 1,600 TWAS associations. Many of them, like 300, were showing association with proteins, which were reflecting of translational processes, which you can see here. Others, like for instance, the one which I'm highlighting here, is showing for, in fact, direct interaction between the prednisol and FKBP5. It was shown before in the literature that the prednisol is causing an elevation in the FKB5 level. So many of those associations were also showing the one which were previously described in the literature by other occasions, not the TWAS. But what was surprising to us was the identification of over 1,000 lipid structures and lipoproteins from the TWAS. So then further evaluation brought us to conclusions that in fact those 1,000 molecules are surrounding only 5 transcripts, which is GATA, HDC, and also FCER1A, MC4A3, and PDK4. Looking closer on those, we saw that there is certain overlap between 4 of them, but not PDK4. So the associations which were found for PDK4 were not overlapping with any of the 4s. So we further check it using the ingenuity pathway analysis, and it indeed also here we found out that the GATA is interplaying with 3 out of those 4, but PDK is not interplaying with those molecules. So we had further the closer look to those processes, and we find out that PDK4, which is pyruvate dehydrogenase kinase, very important molecule, especially when we are talking about a switch between fatty acid oxidation and glycolysis, this molecules is critical for the adaptation of cells, and this is the associations which we see in terms of the fatty acid can be explained in that way. Further looking at the for other, we found that they are mainly associating with thiacylglycerols and VLDL for all 4, but GATA and HDC are associating with LDL, HDL, so they are showing positive associations with those, and also ApoA along with phosphatidylcholines and lysophosphatidylcholines. So given the nature of our study, where we were making the transcriptomics from the blood, of course, we ask ourselves a question from where those gene expression might be coming, and we were looking at different composition of the blood, and we find out that basophils are in fact expressing all of those 4 molecules. So we came to the conclusions that in fact the TWAS revealed the interplay between the lipids and immune responses, which are in fact of relevance, especially for chronic inflammation and arteriosclerosis. And it could be through our cohort, because you need to think that type 2 diabetic are frequently having chronic inflammation. Next, I would like to move to the phenotype and how different platforms are explaining phenotypes such as sex, age, type 2 diabetes, and BMI. And as you can see here is different amount of the molecules correlating with those different phenotypes here. I would like to highlight only briefly the associations with age. So this is only the top molecules which are associating with age. So the strongest associations are with metabolites related to hormonal regulation, which is again very valid point and very relevant, but also glycans, which were before proposed as the markers of age. So our next question, which we asked in this context, was what is the percentage of the variance in each of the phenotype, which can be explained by each platform. And you can see here that clinical platform, so the clinical is kind of reflecting very well on different phenotypes. Also very sensitive is Olink and Soma towards the phenotypes, as well as the 2 platforms of metabolomics which we were using. So now I would like to very briefly focus on diabetes. So we were looking at the molecules which are showing association with diabetes, and we identified 129 molecules, which majority of them were previously linked with diabetes. But I would like to mention here that diabetes is a very heterogeneous disease, and especially latest study were showing that in fact there are 5 different subgroups of adult onset of diabetes. And this was a study presented by Alquist et al. In a Lancet, who based on factors such as HbA1c, BMI, age, HOMA-IR, was able to generate those 5 clusters. And those clusters are defined here. What we were trying to understand, whether or not those clusters kind of associate with specific molecular features. And this question was asked by Shaza, who is a very good friend of mine. So she was looking whether or not similar clusters can be created in Arab population, specifically here in Qatar Biobank. So what she did, she used exactly the same algorithm as the one in the Andes study, and she was able to show that the distribution across different clusters, which we are having here highlighted in different colors, is in fact recapitulating the one which can be seen in Andes study. Next, she was trying to find the association between those clusters and proteins as well as metabolites. And just to give you an example and showing that really the associations are really making sense, the one cluster which was severe insulin resistance can be highlighted as the one with the highest level of the insulin, where the cluster which is mild obesity related is showing the higher leptin levels. I mean, I won't gonna be going through each of the molecule, but believe me, or look to the manuscript of Shaza, all of them, they are really making true sense. She did exactly the same exercise looking at the metabolites. And here again, in the cluster of severe insulin deficient, she was identifying 1,5-HG mannose and glucose as really relevant for this cluster. So she was able to establish the proteometabolic signatures, which were unique to each of this diabetes subgroup. But what we were trying to do using our multi-omics resources, we were trying to understand if we can create the entire network of interactions around the features which she find out for each of this diabetes subgroup. And this is only a very brief example of the MART subcluster, which is characterized by the age of the diabetes onset. So what we did here, we identified 108 molecules which are relevant, and 5 of those we also find 5 of the main cluster here. So it's the especially osteomodulin, fructose, glutamine, and callicarine 7. So I would like to focus your attention on the osteomodulin cluster, because here we were identifying 3 molecules which are proteins, in fact, contributing to arteriosclerosis and plaque formation, as well as calcification, which suggests cardiovascular complications potentially in the MART cluster. Further, glycine, which is known to be a major quantitative and structural component of collagen, which is also contributing to calcification in the context of cardiovascular diseases. And next, cholesterol esters to total lipid in VLDL, which are known to be associated with cardiovascular complications. So basically, the conclusion from here is that the molecular network is providing further insight into the disease pathology, and is suggesting that subjects with in marked subgroup are potentially of risk for developing cardiovascular diseases, but all related to calcification. So I believe that with this, I was able to go through the complementarity, integration, and analysis of the data. But right now, I would like to very briefly focus on visualization. I can see that we are kind of running out of time, but I will try to be very quick. So what I'm showing you here is the way how we are trying to encourage people to look for their protein of interest and how we integrated all of our data to be available for the scientific community. So please have a look on our server, enter our server and look for your molecule of interest. I would like to say here that this server was designed and done by Professor Karsten Sure, and we are trying on his blog to update more on the molecular features and molecular associations, which we are able to see based on our multi-omics data. So only brief example how you can generate hypotheses related to different molecules. For instance, the methyluridine is known to be associated with different inflammations, and this is the molecular cluster generated with comics related to 5-methyluridine. And what we can see here, the two metabolites are reflecting on really metabolic processes of 5-methyluridine, again, supporting the correctness of our data. But we are also seeing around 20 CpG sites near 13 different genes and 4 gene transcripts, which we further evaluated, and we find that they are involved in innate immunity, further suggesting the role of 5-methyluridine in inflammatory processes. Further, we identified cholesterol, which is a signature of rheumatoid arthritis and known to be anti-inflammatory molecule. So all of those together could suggest that 5-methyluridine is potentially having immunosuppressive properties, which of course need to be further evaluated. So again, here is a brief overview on our comics. And again, I'm encouraging you to visit the website. We see comics as a tool to explore further molecular human. I believe that with all of this omics, I kind of convince you that this is the best representation so far of the molecular processes which are ongoing in the human body. And we see this work as a benchmark and kind of stage for future bigger initiative. So with all of this, I would like to especially thank Professor Karsten Sure, who is the main investigator on this study and is amazing mentor supporting and letting me to work on projects like this and part of the fusion between the bioinformatician and biologist. I would like to also acknowledge people from the course, including Frank, Hani, and Joel. Also Jan Krumzik for the work we are doing together and other collaborators across the globe. And last but not least, I would like to acknowledge Metabolon, especially Annie for her involvement in our QBB study and metformin. So with all of this, I would like to thank you for your attention. And yeah, I'm ready to take questions.
Thank you very, very much, Ania, for a wonderful presentation. I would also like to encourage you to go check comics out. I think it's a wonderful complementary tool for people working in the multi-omics space to quickly identify associations between different analytes, different biochemicals. So great job there, Anna, and can't wait to see how it evolves in the future. We do have a couple of questions in the chat. One question, which I believe refers to the first part of your presentation about whether the questionnaires were administered at the same time as the blood draw.
Yes. So in the QBB, the patients or the people which are enrolled to the QBB are undergoing the complex process which is happening during one day. So they are going through the questionnaire, they are going to the blood draw, and all other measurements in the same time. So this is happening in one day time.
Mm-hmm. The next question asks, how can we use these insights from multi-omics to develop point-of-care diagnostics? Is it possible to develop decentralized diagnostic assays from these insights?
So I believe, I mean, it's very good. It's a very good question, because first of all, this kind of approaches, are kind of budget intense, right? But when we are thinking from the perspective, metabolism is not sufficient to give us certain answer. Also, proteomics is not sufficient to give us certain answers. I believe that combination of three different omics levels will be the one which will be sufficient to further develop the assays. But how to do it? First of all, it needs to be specific question asked. What kind of disease you are interested in looking at? Is your omics really showing discrimination between different subpopulation of the patients? So let's say if you were going to be applying multi-omics on diseases which maybe not necessarily will going to be showing a signal, of course, this will be not the right assay. But once you see the signal, you will gonna see in which capacity, which omics is really capturing the best the signal. Then it will be the appropriate step to take next one. So saying, okay, I'm seeing something from metabolomics, I'm seeing something from proteomics, and maybe on the genetic level, to have a toolkit which will be able to really be implemented in the diagnostic. But of course, multiple tests are required. And of course, there must be an objective of the disease, which is not well captured by clinical chemistry. I believe you saw, I showed how well clinical chemistry data are depicting, for instance, type 2 diabetes, of course, not to the subgroup extent, but it's kind of, it's kind of sensitive towards sex, BMI as well. So, the question is what kind of disease you have and whether or not this disease is difficult to determine. Also, responses to the treatment will be another application of multi-omics, because not necessarily clinical chemistry will be the right way to go. So I'm covering it that way.
100% agree with you. So there is a more technical question in the chat about whether you could briefly discuss how you actually integrated the omics data. The question says, it seems you just checked simple overlap among omics, but I'm sure it's not as simple as as it looks.
I mean, it could be considered as simple, but it's not. So basically, we were not integrating them in, like, there are tools like, for instance, MUFA, which I think Dr. Snyder is using. So we were not going that path. We were utilizing different tool, which was this mutual best hit, and it was the first time we were trying to look at mutual best hit in the context of omics. So this was one way of integration. Second was the so this was the integration of between omics, but also between platforms. The second layer was GGMs, so Gaussian graphical models, which were showing the association within given platform. And on top of this, we conducted the GWAS, EWAS, and TWAS. So all of this together, we integrated in one molecular network, which can be right now available for entire society. And I think this is the biggest beauty of this project, because right now everyone is having access to it, and everyone really can search for the favorite molecule.
Mm-hmm. Which, yeah, I think your lab and your network of collaborators are really doing a wonderful job when it comes to making this data accessible for everyone to be able to use and validate, replicate, expand on their own research. And one last question is whether you have considered the possibility of trace levels of certain medicines in the public water systems. Studies have shown
that's a very good question, and that's the question which we were in fact asking, because when you think about the metformin and the level which is showed in the subjects which shouldn't be seen, this is one of the questions which could be asked, that maybe there is certain contaminants. And indeed, we are thinking about study like this. We know that there is a lot of contaminants in the waters from river, for instance, and it was very nice publication. And in PNAS, I forgot the authors, but they were showing that literally every single river they were investigating was having contamination with metformin and also all other possible drugs, which is scary, but also is bringing a red flag for us as a society to start to do something about it, and maybe think about better system to clear up, because all of those medications that the animals which are living with us on the earth are exposed to.
All right. We have one more question, technical question. Are you planning to collect samples during a 24-hour cycle? What if the fluctuation of metabolites, of the levels of metabolites, are important regarding subgroups?
Subgroups of what? Of the type 2 diabetes?
I'm assuming.
Okay,
That's
so
the patient clusters.
Okay, the patient cluster, so subtypes of type 2 diabetes. Well, it will be interesting indeed, because we will gonna know how those individuals are behaving at different stage of the organism. So at the fasting stage and then after the postprandial state. We were not honestly planning such a study, but I think such a study would be of interest. The question again is coming back to the question what you really like to extract from such a study, what kind of message you are willing to achieve. I mean, you will be seeing the differences in the physiology between the normal group and the different subtypes. It's kind of interesting what can come out of this, especially if we are thinking about different medications which people can get. And yes, I mean, would be nice, but I'm more interested in cancer than diabetes. So my work, latest work, is focusing strictly on breast cancer patients, and this is the energy which I'm streamlining right now, especially in context of multi-omics.
All right. Thank you very much, Ania. I do have one last question for you. In your opinion, and sorry for going back to type 2 diabetes and metabolic diseases, but from your experience and everything you have done to date, what do you think would be the wisest selection of omics combination? What will be the best combination with metabolomics plus one or two more other omics to address complex diseases, complex metabolic diseases, so such as diabetes and obesity?
So what we could see on the cluster coming from the multi-omics, so what we did, we took proteomics data and metabolomics data, the one which Shaza was analyzing in those subclusters, and we put them in the context of the molecular network. What we observed was majority of the molecules which we found were molecules of metabolism, and were also proteins. So proteomics for sure, metabolomics for sure, lipidomics for sure. So there are those three omics which are very important, which if I will gonna have, let's say, limited resources and I will be interested in, probably I will gonna select those, especially they can give us extra hints on the processes which are ongoing. And this was the process which I was highlighting here related to calcification and cardiovascular diseases. And I think this is one of the most important things from our finding related to diabetes subtypes, because it can be further follow up. It also will be important from the perspective of using different medication and maybe starting specific medication before a patient will gonna be progressing further into cardiovascular diseases and complications.
Excellent. Thank you very much, Ania. So
thank you very much.
As we closing today's webinar, I just want to thank you on behalf of everyone at Metabolon. We want to thank you and everyone involved in this project. The work that you guys have done has really has provided such an immense value to the field as a whole, and we feel very privileged to have been able to contribute to that. Thank you very much for an exciting and wonderful presentation, and yeah, we look forward to continuing our work together.
Thank you very much. It's a great pleasure for me. As I said, I'm only privileged to show this data. It's a main person behind the scene, I will gonna say, is Kirsten, who was letting me to be involved. And also all other collaborators, especially the one from Hamad Medical, which were working with us, as well as the one from Genus from the perspective of glycation. Of course, Metabolon for me, from my perspective, is very important collaborator because, as I mentioned, Ania Evans, she was strongly involved in our study and she was supporting us a lot, especially on the context of the medication study, the one from the Qatar Biobank, but also the one Gaurav was conducting with the ratios and acetaminophen loci. So I would like to, thank again for the great collaborative effort. I would like to thank you all people which were joining us today. I hope you enjoyed, and I hope that you will gonna be using our server and you will gonna have fun with that, because this is exactly our way of sharing. We want people to have fun from utilizing our resources and doing research, and that's the reason why we're sharing. So thank you for joining us today. It was great pleasure for me being here. Thank you, Natasha, for facilitating this session. It
My
was
absolute pleasure.
Great pleasure. Thank you.
Thanks everyone for joining. The recording will be made available through our website, and we'll look forward to seeing you in one of our future webinars.